Tissue engineering, also called regenerative medicine, refers to the attempt to create functional human tissue from cells in a laboratory. Its ultimate goal is to be a cure, not merely a treatment, by repairing or replacing tissues and organs that fail due to disease, genetic errors, congenital abnormalities, or traumatic injury. Tissue engineering relies on 4 important factors: the right cells to do the job, the right environment, such as a scaffold to support the cells, the right biomolecules, like growth factors, to make those cells healthy and productive, and physical and mechanical forces to influence the development of the cells. The cells can be directly harvested from the target organ, developed from precursor or stem cells, or taken from lines grown in the lab, all ideally from the patient, as that limits problems with rejection. The supporting structures can be derived from donor tissue or from natural or synthetic polymers made to order for their strength or endurance. Biomolecules can be added directly or coaxed from the cells that take up residence on the scaffold. Sometimes the scaffolds dissolve over time, but others remain to provide support to the organ. Some examples of tissues and organs that have already been successfully tissue engineered and implanted in humans include bladders, small arteries, skin grafts, cartilage,
Tissue engineering is the use of a combination of cells, engineering and materials methods, and suitable biochemical and physicochemical factors to improve or replace biological functions. While it was once categorized as a subfield of biomaterials, having grown in scope and importance, it can be considered as a field in its own right. While most definitions of tissue engineering cover a broad range of applications, in practice the term is closely associated with applications that repair or replace portions of or whole tissues: bone, cartilage, blood vessels, bladder, skin, muscle, etc. Often, the tissues involved require certain mechanical and structural properties for proper functioning. The term has also been applied to efforts to perform specific biochemical functions using cells within an artificially created support system, an artificial pancreas, or a bioartificial liver. The term regenerative medicine is often used synonymously with tissue engineering, although those involved in regenerative medicine place more emphasis on the use of stem cells or progenitor cells to produce tissues.
Improving treatment strategies for cardiovascular diseases is a major challenge within the field of regenerative medicine. Here, we induce the regeneration of a blood vessel inside the body by using a cell-free supramolecular scaffold which replaces the injured tissue. In this scaffold, microfibers form a network providing spatial support for new cell growth. These microfibers consist of smaller self-assembled nanofibers, which are formed by stacks of uridopyrimidinones, or UPs in short. Bioactive UP-modified peptides, shown in yellow, are incorporated to provide necessary biological signals to cells, while other UP building blocks are introduced to meet the mechanical requirements and to add anti-fouling properties. Since the self-assembly of the UP groups is reversible, this imparts a dynamic nature to the material. Tailor-made materials can be developed by a simple mix-and-match strategy of customized UP functionalized building blocks. These new materials are characterized by their dynamic behavior, with the ability to respond and communicate to the surrounding native tissue. For example, biological signals at the luminal side of the scaffold selectively recruit cells from the circulating blood. Here, peptides assembled with the scaffold direct the cells to differentiate into the desired phenotype. As a result, endothelial cells will cover the luminal side of the scaffold and are surrounded by vascular smooth muscle cells in the medial layer. Due to the dynamic nature of the scaffold, formation of the new vessel is accompanied by gradual resorption of the scaffold material, ultimately resulting in a fully regenerated tissue resembling the native blood vessel. Supramolecular materials form a new and promising generation of smart materials within the field of regenerative medicine. The mix and match strategy applied here allows for tuning of the material properties towards application-specific demands, not only for replacement of coronary arteries, but also for the development of heart valves and peripheral blood vessels.
Goals of tissue engineering. Tissue engineering evolved from the field of biomaterials development and refers to the practice of combining scaffolds, cells, and biologically active molecules into functional tissues. The goal of tissue engineering is to assemble functional constructs that restore, maintain, or improve damaged tissues or whole organs. Artificial skin and cartilage are examples of engineered tissues that have been approved by the FDA. However, currently they have limited use in human patients. Regenerative medicine is a broad field that includes tissue engineering but also incorporates research on self-healing, where the body uses its own systems, sometimes with help foreign biological material to recreate cells and rebuild tissues and organs. The terms tissue engineering and regenerative medicine have become largely interchangeable, as the field hopes to focus on cures instead of treatments for complex, often chronic, diseases. This field continues to evolve. In addition to medical applications, non-therapeutic applications include using tissues as biosensors to detect biological or chemical threat agents, and tissue chips that can be used to test the toxicity of an experimental medication. Importance of tissue engineering. Tissue engineering is a radically new concept for the treatment of disease and injury. It involves the use of the technologies of molecular and cell biology, combined with those of advanced materials science and processing, in order to produce tissue regeneration in situations where evolution has determined that adult humans no longer have innate powers of regeneration. Tissue engineering, however, along with some other aspects of regenerative medicine such as gene therapy, has yet to deliver real successes in spite of a considerable science base and investment in the commercial infrastructure. This article addresses the underlying issues of benefit and risk in tissue engineering in an attempt to understand why this situation has developed. There is an inherent, virtuous logic to tissue engineering that sounds too good to be true. By my definition, tissue engineering is the persuasion of the body to heal itself, achieved by the delivery to the appropriate site of cells, biomolecules, and supporting structures. It specifically involves the regeneration of new tissue to replace that which has become diseased or injured, the significance of which is that we, as adult humans, do not normally possess this ability. We may repair ourselves under some very limited circumstances, for example, bone fractures and injured skin may undergo repair, but, even when this does occur, this usually involves non-specific reparative tissue, scar tissue, rather than the regeneration of the specific functional tissue that has been affected. The essence of tissue engineering is that those cells capable of initiating and sustaining the regeneration process are switched on, perhaps through growth factors or genes, so that they generate new functional tissue of the required variety. This may be achieved with the help of a scaffold or matrix to guide the geometrical or architectural shape of the new tissue, and may take place on a customized basis at the site of the injury in an individual patient, or on a more industrial scale in an ex vivo bioreactor, where the resulting tissue construct is reimplanted into the patient. Issue in tissue engineering. Tissue and organ failure, produced as a result of injury or other type of damage, is a major health problem, accounting for about half of the total annual expenditure in healthcare in the U.S. Treatment options include transplantation, human or xenotransplantation, surgical repair, artificial prostheses, mechanical devices, and in a few cases, drug therapy. Ultimately, however, major damage to a tissue or organ can neither be repaired nor long-term recovery effected in a truly satisfactory way by these methods. Tissue engineering is emerging as a significant potential alternative or complementary solution, whereby tissue and organ failure is addressed by implanting natural, synthetic, or semi-synthetic tissue and organ mimics that are fully functional from the start, or that grow into the required functionality. Initial efforts have focused on skin equivalents for treating burns, but an increasing number of tissue types are now being engineered, as well as biomaterials and scaffolds used as delivery systems. A variety of approaches are used to coax differentiated or undifferentiated cells, such as stem cells, into the desired cell type. Notable results include tissue-engineered bone, blood vessels, liver, muscle, and even nerve conduits. As a result of the medical and market potential, there is significant academic and corporate interest in this technology. Advantage and disadvantage of tissue engineering. The pros of tissue engineering are obvious. Engineering tissues can potentially help a person conquer a disease or illness. Tissue engineering may even be able to cure extreme and mild arthritis in patients that receive this sort of treatment. Tissue engineering also has the capability of prolonging our lives and making the general quality of our lives much better. The cons of tissue engineering are fewer than the pros, but they are present. Two main cons to tissue engineering are often discussed. One is the possible presence of latent or hidden diseases or illnesses in the base tissue. There is the possibility that if foreign body tissue was used to reconstruct particular tissue matter for a patient, there could be hidden disease or other hidden illness within that tissue matter. This is something that scientists are working hard to prevent. However, latent diseases are troublesome to find with the technology we have now,